Meiosis I, cell division. The parent cell, oops, there should be two red chromosomes here. There's replication of the chromosome with meiosis I and with mitosis, the similar process. In prophase, the two sister chromatids line up together. In prophase I of meiosis, the tetrads line up together. So there's a synapsis of homologous chromosomes. In metaphase, the chromosomes align at the metaphase plate. In metaphase I of meiosis, the tetrads line up on the metaphase plate. And then anaphase and telophase, the tetrads, the homologous chromosomes separate, but the sister chromatids are still together. You end up with a haploid, or n=2 cell. Here you start with what should have been 4 chromosomes, and here you have 2 chromosomes in each cell. Comparing to mitosis, where the sister chromatids separate at anaphase and telophase, and you have a diploid cell. 2n=4, 1, 2, 3, 4, and the sister chromatids have separated. Meiosis II, the second phase of meiosis, there's no further chromosomal replication. You start out with the 2 haploids, daughter cells from meiosis I, and they undergo prophase, metaphase, anaphase, and telophase as a second division, and we end up with the sister chromatids separating, and you have 4 cells that are haploid. So you start with the 1 parent cell that's diploid, and you end up with 4 daughter cells that are haploid. The key to genetic variation is 3 things: independent assortment, crossing over, and random fertilization. In independent assortment, there are a couple of possibilities. In metaphase, meiosis I, either one of these arrangements is equally probable. We could have the green chromosomes large and small on the left side and the blue chromosomes large and small on the right side. Over here we have the green large and the blue small on one side, and the blue large and the green small on the other side. They're equally probable to happen. During metaphase II, when the chromosomes line up again and the sister chromatids separate, we end up with a variety of possible gametes. Here we have the large green and the small green, large blue and small blue, large green and small blue, and large blue and small green. Crossing over is also important in genetic variation, and this can occur during prophase, of meiosis, prophase 1 of meiosis. Here we have the two chromatids, and from two different kinds of chromosomes. This is the tetrad with homologous chromosomes and sister chromatids. Here they're crossing over on the end. You notice on the green chromosome we have capital A's and capital B's, and on the blue chromosome we have small a's and small b's, and they're crossing over at the bottom. After crossing over is completed, we have the capital A's and a big A, big B, and a small b. The blue B has attached to this set of chromatids. And conversely, the green large B has attached itself to this set of chromosomes. So that's one way to get unique genetic variation in new organisms. Random fertilization is the third type of key to genetic variation, and that simply is that there's a random joining of two gametes. You have the female and the male, each contributing one gamete to the fertilization. There's 2 to the 23rd, since there's 23 pairs of chromosomes possibilities for the female, and 2 to the 23rd possibilities for the male. So there's 64 trillion combinations possible between just one set of parents.